Hi, everyone. My name is Remy Schneider. I'm a postdoctoral researcher in the Snoke Lab. And today I'll be discussing the imaging of human lung organoids with light sheet microscopy. So first, a bit on lung development. The respiratory system originates from buds on the ventral anterior foregut endoderm that emerge around week 4 in humans from lung endoderm. These buds will undergo branching morphogenesis in the stalks, which will become airways, while the distal tips widen into primitive alveoli. Mature alveoli contain alveolar epithelial type 1 cells, which are essential for gas exchange, and alveolar type 2 cells, which produce surfactant, crucial for maintenance of alveolar integrity. Directed differentiation attempts to recapitulate this lung development through generation of lung organoids that correspond to the second trimester of human development. So firstly, definitive endoderm is induced from human pluripotent stem cells in the presence of high concentrations of activin A. Subsequently, lung-biased anterior foregut endoderm is specified by sequential inhibition of bone morphogenic protein, transforming growth factor B and Wnt signaling. Anterior foregut endoderm is then ventralized with a variety of cytokines, including CHR, which then activates Wnt signaling. And finally, lung bud organoids in suspension are embedded in matrigel to initiate their branching morphogenesis. And on the right here, we can see images representing that. This approach allows us to obtain progenitors that express markers of alveolar and airway epithelial cells, a majority of which can be identified as alveolar type 2 cells. The Snoke Lab has developed these 3D organoids as a model of Hermansky-Pudlak syndrome, as well as human respiratory viruses. So to rigorously analyze these organoids, it's very important to understand the spatial distribution of lung progenitors to evaluate organoid maturity, particularly to distinguish proximal airways and distal alveolar cells, and of course to monitor reaction to disease. So the ability to do this can be limiting when using 10 to 20 micron cryosections and a standard confocal microscope. So light sheet fluorescent microscopy allows imaging of larger tissue, of course, including lung organoids and mouse brain, et cetera. And it illuminates the sample with a thin light sheet that lies in the focal plane of a detection objective. This sheet illumination is faster than point scanning of a standard confocal microscope, so low pixel dwell time and reduction of photobleaching results. Taken together, these are some of the features of light sheet that allow for imaging of these larger samples. Additionally, advances in tissue clearing allow for deeper penetration of laser light into complex and dense tissues. Organoids in particular have a high density at their core. So we use the clarity method with some adaptations. So in A here, you can see a cell in its native state. In B, primary amines allow the cross-linking of proteins in DNA using paraformaldehyde and monomeric acrylamide. This is followed by acrylamide hydrogel polymerization to develop a rigid scaffold on which our biomolecules of interest can be held. In C, we see delipidation with SDS. SDS inserts itself into the lipid bilayer, destabilizing it and incorporating these lipids into a detergent micelle that's later carried away during wash steps. And finally, refractive index matching, which renders the sample translucent to increase this light penetration. This is the workflow. I won't go into too much detail for this particular talk, but the key features of the process are the delipidation. It must be very thorough to remove lipids that scatter light and cause spherical aberration during the imaging. Also, the hydrogel must be the appropriate composition to allow penetration of our reagents, particularly the antibody cocktails, but still remain rigid enough to support the structure of the organoid. And so the uniqueness in this particular methodology that's been adapted for lung organoids lies in the retention of a hydrogel sort of encasement, and also in the method used for mounting the sample onto a confocal dish in the Leica digital light sheet SP8 system. And not only is the hydrogel polymerized throughout the organoid itself, there is this protective shell, and both of these are working to retain the complex and fragile branching architecture. The sample is mounted upright with silicone, so there's enough height for the mirror-generated light sheet to easily pass through the sample and for the instrument to be properly calibrated. The sheet of light is digital because the light source emits 2 points of light that hit the mirror. They move rapidly across the face of the mirror, creating the light sheet. So onto some images here. So these particular images represent a 1.4 millimeter cube section of a lung organoid that's been imaged. And mucin-1 secreted by goblet cells in the lung epithelium rather, can be identified here on the apical side of a branching lung organoids and in the lumen there. So it's nicely shown in this particular manner. And this is also accompanied by EpCAM denoting epithelial cells. When we look at two-dimensional standard confocal microscopy of maybe 15 micrometer cryosection, we don't get as much information. And moving forward, here we see two-dimensional images of NKX 2.1, transcription factor and marker of lung progenitors, and vimentin, which is classically a mesenchymal marker. That's in 2 dimensions. So in 3 dimension with the light sheet, we can garner a bit more information. So interestingly here, vimentin is expressed at the tips during partial epithelial to mesenchymal transition when epithelial cells undergo a shape change while in the process of budding and branching, they do deposit vimentin. So 3D imaging allows us to visualize this. Moving forward, so collagen 4 here can be seen. It's a basement membrane protein expressed by AT1 cells. Single cell RNA-seq has shown that these organoids do not contain AT1 cells. However, the stalks in these images do show elevated deposition of collagen 4, but the cells that express this are unknown and still under investigation. But light sheet allows us to visualize the spatial distribution of this particular marker. But however, the surfactant protein C, which is produced by AT2 cells to lower the surface tension in alveoli, is seen here, primarily expressed at the distal tip. So overall, this type of information, it allows us to see cell type heterogeneity between the proximal stalks or the core and the distal tips, which almost exclusively here express a marker of alveolar progenitors. So in summary, by maintaining the structural integrity of a fragile and complex organoid, rigorous clearing methods can follow to maximize excitation light penetration. And also light sheet fluorescence microscopy offers a way to identify a cell type in 3-dimensional space and understand its relationship to the whole. And these are my acknowledgements. Thank you for your attention, and I'll take any questions at this time.